Hello, everyone. Welcome to Creative Biolabs live webinar series. I would like to introduce Dr. Sarah Voglan, who will briefly present our company's gene therapy platform.
Thank you for the introduction, Kathy, and welcome everyone. I would like to start with a brief introduction of our company's relevant platforms, such as our cell line-based gene editing and CRISPR-based services. To start off, our cell line-based gene editing services entail a concise workflow, which includes assessment of the target of interest, transfection of the cells using CRISPR tools, followed by generation of single-cell clones and verification of these single-cell clones by PCR and genome sequencing. The scope of our services include gene knockouts, knock-ins, point mutation, and customized cell line development. This service can help overcome the limited availability of patient samples, which is often the case, and provide stable and consistent models. In addition, we can provide great flexibility in terms of the required genetic modifications as well as cell types, and our services are scalable for both research and production and come with excellent support and affordability. Our CRISPR-based screening services, such as, for example, a CRISPR knockout screening, as you can see on the slide here, involve engineering of the target cells to ensure they express the Cas9, followed by quality control using RT-qPCR. Using the proper library virus, we will then determine the optimal multiplicity of infection, which will be followed by functional screening and data analysis. Sequencing of the cells after treatment will reveal the genes that influence the phenotype of interest. The scope of our services here comprises different types of CRISPR, including knockout, activating, and interference, and customized CRISPR screenings. Our services again feature high specificity and precision and are compatible with various library scales, can provide flexible screening strategies and rigorous QC and advanced data interpretation. In addition, we provide multi-layered readouts, affordable pricing, and excellent support. If you are interested in any of these services or related products, please feel free to reach out to us at the indicated phone number here or email us at info@creative-biolabs.com. And with that, I would like to hand the floor back to Kathy, who will now be introducing our speaker.
Yeah, thanks, Sarah. In this section, we are honored to have Dr. Omar Abudayyeh guide us to explore these transformative technologies and their implications for the future of medicine and biotechnology. He will share insights into the latest developments and future directions in this rapidly evolving field. Now, let me introduce our speaker for today, Dr. Omar Abudayyeh. Omar Abudayyeh is an assistant professor at Harvard Medical School and the director of Abudayyeh Gutenberg Lab. This lab focuses on developing gene editing and synthetic biology technologies through protein engineering and AI, with applications in therapeutics and aging research. He has pioneered CRISPR innovations with dozens of patents and over 35,000 citations across more than 50 publications in top journals. Before Dr. Omar Abudayyeh begins his presentation, I want to quickly remind our audience that we have a Q&A section at the end of the presentation. And if you have any questions during the presentation, please type your questions at the Q&A panel, and Dr. Abudayyeh will get to them after his talk is finished. Now, let's welcome Dr. Omar Abudayyeh. Dr. Abudayyeh, thank you for presenting your important findings today, and you may begin when you are ready.
Great. Thanks so much for the wonderful intro. Let me just pull up my slides real quick. Is that coming through?
Yes, we can see the slides.
Okay, perfect. Great. I'm going to talk a little bit about sort of a few different projects in the group around trying to build programmable technologies to both study and treat genetic disease, as well as some of the aging work in our group. So maybe just to dive in, a question we like to ask a lot in the lab is biology really programmable? And if we could sort of figure that sort of puzzle out, what could we do with that? And I think, we're all familiar with these types of, posters and complex diagrams of what makes a cell and pathways and circuits. And when you see this, it can, seem very overwhelming about how one could actually break down reprogramming cells. But I think, the way we think about it is at different layers of programmability. So a lot of our time has been spent on proteins and molecules, for example, using RNA-guided proteins for all sorts of manipulations, DNA editing, RNA editing, epigenetic editing, even, sensing proteins and protease-based, cellular manipulation. And so, um, that kind of leads into, moving beyond molecular to thinking about whole cell states. And, with sort of the advent of single cell sequencing, really understanding that cells can be very unique and different in subtle ways. And, there might be ways to actually manipulate those states programmably. And then lastly, thinking about like whole organisms and why, people develop and age, and, that process is very different between people. And if we could really try to understand that and reverse that, that would be powerful. So, um, our work sort of falls into a few buckets of genome editing and protein engineering, um, ways to sort of reprogram cells using different types of genetic perturbations and libraries, as well as modeling all that data with AI. And I'll talk a little bit about that, like in terms of virtual cells, and then going, at the full organismal level of trying to really build precise, for example, molecular clocks, single cell clocks, and even modeling that data for sort of virtual level human predictions. Um, so just to dive in at the molecular scale first, one of the new technologies from our group we has been focused on RNA writing. So the idea of, can we rewrite RNAs programmably the same way we can do base editing or prime editing? Um, and I would say over now, I don't know, I guess the past 10 years, the DNA toolbox has gotten very mature. We've gone from just blunt, scissors with CRISPR and for nuclease-based editing to more atomic level precise technologies like base editing and prime editing. But with that tremendous progress, it doesn't feel like the clinical side has really kept up. There's 7,000 genetic diseases, millions of mutations that can drive those diseases, and yet we have one approved CRISPR therapy for sickle cell disease, and works with thalassemias as well from CRISPR and Vertex. And maybe you could argue Intellia is maybe close behind with the first in vivo gene editing drug, hopefully approved in the next couple of years for amyloidosis. And there's still thousands of diseases left untreated, and there is exciting stuff coming down the pipe, but it's not maybe as fast or as scalable as we'd like. And one thing we also have to realize is a lot of these diseases are actually driven by many mutations, and a lot of these drugs coming down the pipe, especially base editing and prime editing, only target one of the mutations. And so there's still, even when you might say a disease is cured in quotes, it's you have to ask the question, cured for who? And one thing we also like to think about is are there new types of edits we could make that could solve some of those problems? And while most edits can be made now, one of the big things missing still, and now over the past few years from us and others, has been somewhat solved, but still has more work to do, is large insertions. So thinking like, we can do, basic cuts and indels, small insertions, small deletions, but when you start getting into the hundreds of base pairs or thousands of base pairs, a whole, thinking about replacing entire genes, that becomes difficult in non-dividing somatic cells. And so there's a lot of work thinking through enzymatic approaches for doing this with transposases, integrases, recombinases, and so on. Just to drill home the point, this is an example of the sort of distribution of mutations for different, I think this is for liver and eye diseases, but basically showing that most diseases are not a single mutation. There's actually dozens, hundreds, in the case of cystic fibrosis, thousands of mutations in the patient population that can contribute to the disease. And so if we really want to think about how to solve this problem, one approach could be, instead of targeting the mutation, right, is targeting the gene. So going to the gene level of treatment. And so if you want to think about replacing parts of the genes like exons or entire genes, you actually need technologies that can do enzymatic gene insertion or gene replacement. And so one of the technologies we developed over the years, we had a paper out, and so did David Liu, was using CRISPR and prime editing in combination with integrases. Integrases are great enzymes. They can load tens of thousands of base pairs into a genome. They're just hard to reprogram. And so we spent time basically using the prime editing system to lay the target sites for these integrases in the genome, and then doing an all-in-one insertion of a large payload. And that actually works pretty well. But there are challenges with that approach. Maybe I'll just go back for a sec. Um, One of the challenges is you have this large protein, you have two guide RNAs, you need the DNA payload as well. And so you have this multi-component drug that you have to develop. Maybe you make these into RNAs, right, and package these in lipid nanoparticles, but the DNA needs to get into the nucleus, and nuclear delivery with nanoparticles is difficult. And so this likely has to be an AAV. So you're thinking about, a dual AAV LNP drug system that you have to figure out how to manufacture and how to get it into the right cells in the body. And it's doable. One of the companies we had spun out, Tome, put out a preprint last year that we could get 50% gene insertion for PKU in the liver in non-human primates, actually. But one thing our lab likes to think about is, can we keep pushing the bar for what we can do with gene insertion with simpler modalities? So one of the approaches, we went back to the drawing board thinking, well, integrases and recombinases, they're always going to require complex systems. And so we thought maybe there are other mechanisms in the cell we could leverage to still accomplish the same goal. And so one of the things we started playing around with was trans-splicing. So cis splicing is what I'm sure most people are familiar with, where a gene, right, splices between one or more exons and forms a mature mRNA by cutting out the introns. Trans-splicing is where you have a trans template that has a sequence that you want to edit, the edit sequence essentially. And we bind in an intron of a pre-mRNA. And if you have the right regulatory signals on this, it can look like, the downstream exons for the gene. And so in a way it confuses the splicing machinery. And so the splicing machinery has to decide, do I go into this new exon down here, or do I go to my normal downstream exon? And in this case, if you design your templates correctly, as I'll show, you can get fairly high efficiency into this new trans template. And so then you can have an edited mRNA where you can have one or more exons now replaced in your mRNA. And we call this precise. And as I mentioned, a lot of people are like, oh, they want to just write this off because it's already editing. Why would you want that? But I would make the case, there's a few reasons why this actually can be advantageous. For one, RNA, if you look at the regulatory landscape for DNA editing, it's a lot slower because there's just a lot more concerns around permanent edits. And so it can be longer and more expensive to develop those kinds of drugs. But two, by leveraging an RNA writing system that's, as I'll show, protein free, so it's literally just a trans template, we can leverage AAV, which is difficult to do with CRISPR. And with AAV, you can get to a lot of other tissues than liver, like CNS, eye, muscle. There's data coming down the pipe for kidney. And so it actually allows you to start going after diseases that, we could only dream of with LNP-based genome editing. And so we think this technology provides a really differentiated angle for targeting or making a dent in the 7,000 genetic diseases that are yet to be treated. And at the same time, because it's exon and sequence replacement, these therapies are not limited to like rare mutations. You actually can group most of the patients of a disease together. So just to dive in, when we first started playing around with this, we didn't really know what we were doing. So we wanted to sort of improve the chance of success as much as possible. So we thought we could, start to get some signal on the board, so to say, by using Cas711, which is an RNA cleavage system we had developed previously, to just cleave the intron. What we wanted to see is if we cleaved the intron and got rid of the cis exon, could we basically force the system to use our trans template. And when we did that, we actually saw very nice results. So normally one of the things that's limited trans splicing, there's been a lot of groups working on it, has been it's very inefficient. And so we wanted to boost the efficiency so we could start to optimize the trans template. So what we did, we have two readouts here, protein correction and RNA editing. Protein correction is by measuring luciferase signal. We had a luciferase reporter that had a stop codon in it, and then if we have sequence replacement, we can fix the stop codon and see luciferase activity. And we can also sequence the RNA for that and look at the RNA editing. You can see that the protein levels in RNA editing match very well in this experiment, showing that it's all connected. And we had 3 different guides and a non-targeting guide. The non-targeting guide basically tells you what your baseline trans-splicing activity is on its own. So you can see we can get a little bit with natural trans-splicing. It's like 1%, barely detectable. When we cleave with Cas711, we actually get this huge boost, 7 to 9-fold boost in activity. So it really puts us on the board for being able to now optimize the trans template and see how we can get natural trans-splicing better. We also wanted to see if this didn't apply just to luciferase, but we tested a bunch of endogenous transcripts. We also used hyperactive versions of the Cas711 as well, shown with the mutations. And you can see that across different expression levels of the transcript that we were targeting, we can get varying levels of activity. The higher the target was expressed, the editing goes down because you have more molecules you have to edit. But in general, we could get between 5 to 40 something percent. So very nice editing levels. And we could look at these endogenous proteins and also show that we're actually getting protein level editing. So we can install like a flag tag or HA tag on them when we do the edit, and then look at it on a Western blot and see that we're actually making the protein we expect. And then just another sort of aside, when we look at sort of transcriptome-wide sequencing, we don't see any off targets as well. The only significant edit we see is the on target over here. So with that said, that's kind of how we established the initial system. And then we started asking, could we start optimizing the trans template to basically get protein-free editing? And I won't bore everyone with all the details of the engineering we did. You can check out the preprint. There's many figures of, different splicing signals and branch points and, exonic splicing enhancer sequences and, playing around with, stability sequences as well. But one of the main things that actually worked very well is we had suspected that the poly A signal was leading to export of our trans template because you get polyadenylation and then export to the cytoplasm. And so we wondered if just cleaving off the poly A either with Cas711 or with a ribozyme that naturally cleaves off the poly A of the trans template could we basically force nuclear retention for the splicing. And when we did that, we actually found that we could get very nice editing. So shown here is the Cas711 version, targeting and non-targeting guide, and then here's without any cleavage of the poly A. And then here's different ribozymes. And we find that, with the twister ribozyme, for example, we can basically get almost 40% editing with a protein-free, just a single trans template coming into the cell. And also for 2 other genes, we also saw a similar phenomenon where you get fairly high efficiency with just the ribozyme cleaving off the poly A. And what's nice about this is this template is very small. Most exons are, 200 bases. And so you can easily fit this in an AAV where you basically just have a promoter, the template, and the rest of your poly A and template sequence. And for example, this data here, the Huntington's data, is very cool because it's exon 1 for Huntington. So we're basically replacing the exon that gets expanded. And it doesn't really matter how expanded it is or how variable it is, because we bind in the intron downstream and then just replace it. So our system never has to worry about what the exon 1 even looks like, which is in contrast to typical gene editing systems where you have to target the repeats or cleave around them, and it's a difficult process. So for AAV, we're able to actually package a single template AAV and also get still efficient replacement of Huntington's exon 1. And we even did an iPSC-derived human neurons of AAV and could get up to 50% when we sorted for AAV transduced cells. And then we've recently been doing in vivo experiments, which I'm not ready to share yet because it wasn't in the preprint, but we're able to actually get in vivo brain editing with these AAVs as well. So we're pretty excited for. But we feel like we're able to move pretty fast. Typically when you're developing a complex gene editing system, you spend months, if not years, optimizing the components for good in vivo delivery. Here, from the first experiment, we got editing in the brain, and we'll continue to be able to optimize that. So that's the advantage of a simpler system for editing. So as I mentioned, next steps is we're continuing to actually optimize the technology. We're now applying it to different CNS disorders, and particularly Huntington's and Rett syndrome have been 2 that we've been focused on. and really leveraging, as well as I mentioned here, like the new capsids coming from AV companies, where you can get deeper and broader brain transduction from systemic injections, going after kidney genetic diseases as well, I think is really compelling. So there's a lot of exciting stuff to do here. So on that note, I will move to another exciting story we published last year in Science on basically using sort of the advancements in protein language models and AI models for proteins to make better proteins, essentially. So just to dive in, uh, protein language models have come out over the years to model hundreds of millions of protein sequences. And the challenge has been they get good at predicting, protein folding, and people make generative models to do protein de novo design. But typically, you don't get better than wild type activity. You don't really get things that are 10 times or 100 times better. They just barely work. And so we wanted to build models on top of the protein language model that could actually understand protein activity and optimize it so the sequences we're testing are actually much more effective. And so to do this, we developed a system where you basically can use the protein language model to predict sequences to go test. You test those sequences in conjunction with this sort of top layer model that picks the sequences. It picks them and you go test them. And then you feed that data back into the model and it tells you the next set to test. And we do it in a way that we only need like 10 to 20 data points per round. So it's very low end directed evolution, but we find that we can then converge very quickly to high activity sequences. And just to show some high level results before diving into what we did, here, each dot here is a different protein. So 12 different proteins that we have mutagenesis data on. And we simulate our approach, directed evolution, versus pre-training and versus sort of zero shot de novo prediction. So zero shot, most of the mutations are not high activity. With pre-training, you can get pretty good. But here, without any pre-training and just doing 5 rounds or 100 rounds of directed evolution, we can get pretty good results without ever having to do any pre-training. And here, we're getting better basically than 500 sequences that you pre-train on. And the cool thing is we were actually able to evaluate different foundation models for this task, and ESM2 came out to be the most effective. And this is just showing the directed evolution, process for 4 different proteins from round to round. The green line is Evolve Pro, and then the orange and blue lines are with foundation models that are not sort of LLM based, so very basic foundation models. And then the dotted line is zero shot ESM2, and the brown dotted line is random sampling. So you can see here we can get to very high activity variants fairly quickly in just testing 16 sequences per round. And the low end aspect here is important because directed evolution typically requires a lot of work, testing hundreds of thousands of mutations, expensive screening setups that take a long time to set up, a long time to run. And here you can take any assay, any arrayed assay. It doesn't have to be amenable to pooled screening. And it can even be more than one arrayed assay that you can do at the same time, allowing you to basically do directed evolution on anything that you desire, which is a very powerful framework. Just to give an example, one of the things we tried was improving a T7 RNA polymerase. And so T7 RNA polymerase is very important for mRNA therapies because that's how you make the mRNA. And so we thought maybe we could evolve it across 3 different parameters: the RNA yield, the RNA translation in the cell, so how much protein does the mRNA produce, and then the immunogenicity, using an immunogenicity assay of the produced mRNA. And so what we did is we ran Evolve Pro with only 10 mutations per round, and we found very quick improvements. This is the fold improvement across the 3 different assays, where we're getting on average a 15-fold improvement in the protein. And this is blowing it up on 2 different axes: the translation of the mRNA and the immunogenicity. And so you want to go up and to the right. You want higher translation and lower immunogenicity of the mRNA. And so by the end, the best construct is this triple mutant that has 500-fold less immunogenicity for its produced mRNA and up to almost 60-fold higher translation from its mRNA. And just to show, if you look at the fitness landscape of the language model versus activity landscape learned by Evolve Pro, it's very different, which is why you can't, do zero shot prediction well. Another application we looked at was therapeutic antibodies. This is a SARS-CoV-2 antibody that Regeneron made that was already very, very high binding activity, and we wondered if we could just improve it with Evolve Pro even better, and we were able to get it twofold better binding activity for something that was already low nanomolar binding. And these are two other antibodies that we evolved from scratch. One is a COVID C143 antibody, and another one's a transferrin antibody, CD71. And you can see here in just 5 rounds, we're able to get improvement not just on binding, but also expression. For example, the CD71, we get 10-fold higher expression and almost 30-fold higher binding. And in both cases, the antibodies are low picomolar binding affinity with Evolve Pro. So we're able to get very strong binding. So in general, we're very excited about Evolve Pro for therapeutic development. One thing we're focusing a lot of time on is antibody development. So where you basically can feed in a target product profile for the antibody, like I want longer half-life, I want better sweeping characteristics, or maybe better FC receptor engagement, better solubility. You can feed all of that data into the model and just spits out sequences that optimize along those axes. And also just, in general, using this for bispecifics and trispecifics, which are traditionally hard to make. So, that's kind of our foray into using different ways to program molecules. So now I'll transition to basically some of the sort of cell-based and organismal-based modeling that we do towards aging and some of the AI work we're doing in that space as well. So, one of the things we like to think about a lot is how can we rejuvenate and reprogram cells, and is it even possible? And to do this, one of the things we typically think about is, first we need to define aging at a molecular level. We typically think of, single cell omics as a way to do that. Once we can do that, we can then use perturbation screening with these single cell omic readouts as sort of the readout for the screen. And then we can use all that data to do additional modeling and prediction. Traditionally, molecular level readouts in aging have been focused on sort of whole blood and tissue level, readouts like epigenetics or proteomics. But if we're going to do, molecular screening where we, for example, perturb thousands of genes at once, we need single cell resolution versions of these. And so we spent a lot of time developing low-cost single-cell preparation methods that can allow us to do perturb-seq screens, essentially, where we can screen, genome-wide knockouts or whatever for low cost. We can essentially do a genome-wide screen now for $20,000. And one of the places we started was with hematopoietic stem cells. So we've taken human donor HSCs from 6 decades of life, and we can do single-cell RNA sequencing on them. And this is just a preliminary cohort of N of 1 person per decade, where we can see separation of the ages, and we can actually build a machine learning clock that can estimate age based on the RNA transcriptomic levels. There's a lot of biology to glean from these clocks. We've only just started, but there's a lot of different signaling and developmental markers that pop out that are interesting and change with age. For example, one of the really interesting ones is SIRT1 goes up in these HSCs. And we found data in the literature that if you actually knock out SIRT1, you can increase the number of lymphocytes produced in the blood. And myeloid lymphoid skew is a classic thing that goes wrong in aging, where you have way more myeloid cells than lymphoid cells, and that's why your immune system declines with function. And so this is just an example of one finding that could have a downstream action item to correct. We also find many other trends, different genes that go up and down, or some genes that go up and stay. There's a lot of nonlinear dynamics that we're also trying to understand here that's pretty interesting. So with that said, with these clocks in hand, one thing we're really excited about is using those clocks as a readout of screens. And so a thing we're doing in the lab now is across different age cells, we can basically do perturbation screening. And one of the ones we like the most is the transcription factor screen, because we find transcription factors are very perturbative compared to CRISPR knockouts. And especially for rejuvenation, transcription factor screening makes a lot of sense. And we published this paper with Feng a number of years ago for building a transcription factor atlas, where we can study the effect of every transcription factor in embryonic stem cells and what cells they cause differentiation into. And we're basically reusing this type of screening approach, but now for aging signatures. And just early stuff coming out is things that can control circadian rhythm, development, stress activation, and even cell cycle regulation. So we're very excited to continue digging into this biology. We also are very excited about the secretomes. This is another type of library that I won't share data on today, but we basically can do pooled screening of every secreted factor in the human genome. which is a really powerful approach for perturbing cells. We basically say bottom up and top down. So transcription factors are like bottom up from the genome, and just the secretome allows us to do top down from like receptors and signaling pathways. And so one of the things we realized is we're collecting all this high, really large scale single cell data across millions of cells and perturbations. Could we actually use this data to build better predictive models of biology? And one of our motivations for this is, we spend a lot of time in the drug development world through consulting and whatnot, and it's pretty disappointing to see how much the rates of success have come down over the years. It's harder than ever to get drugs approved. Part of that's the biology and understanding why drugs might have tox in people, or why they don't end up working, even though preclinical models say they'll work. And so we thought, can we use AI to accelerate every part of the drug development pipeline, from R&D, target hypothesis generation, to screening and in vitro and in vivo models, to even clinical trials. And I would say part of the reason here to do this is if you could basically have models that can improve the predictive capability of your preclinical data, even if we had a few percentage points, the success rates, that could be huge. And it was pretty interesting, like last week, the FDA announced that they're really going to start supporting AI models in FDA packages for this purpose. And so I think, even the regulatory side is interested in moving away from animals and more towards in vitro human models and predictive models that can connect that to clinical outcomes. So there's a lot of tailwinds here. And so what that looks like is, we're building models at every scale here, from reasoning models to virtual cells and tissues to even clinical, virtual humans. I'll focus on our virtual cell work today, where, basically we're building something that can do a lot of different functions. A lot of people ask what a virtual cell is, and, our version of it is something that can, be a reference atlas. You can embed cells and see what they look like. Predicting evolution and dynamics, predicting the effect of perturbations is a big one. Even allowing for new cell states to be generated, or building this up into a digital person would be really powerful. And the next question is, well, then how do you do that? There are some people who might say you have to simulate every molecule, every atom. People are trying to do that. It's very hard. Maybe quantum computers can unlock the compute needed for that. But the other approach is to just take a sort of heuristic approach. Well, there's data like genomic information, single cell RNA-seq, multi-omics, imaging, and so on. Could we get a picture of what a cell is by just feeding all of that into these models? And so we spent a lot of time focusing on single cell RNA-seq. There's been an explosion of single cell RNA-seq over the past 15 years. I'd say 15 years ago, there was only a single cell sequence, and now we have hundreds of millions, and, we're trying to get to billions. And as that data scales, there's a huge opportunity to use it for this type of purpose. Just to put in context, language models like, ChatGPT have tens of trillions of words in these large 50 terabyte, compressed data sets of the internet. Image models like DALL-E are billions of images and hundreds of terabytes. And transcriptome data, if you look at high quality human data, is probably 16 million cells in just a single terabyte, basically. So we have a long way to go to essentially scale the data to get to a point where we can see performance in other domains. But with that said, before GPT-4, there was GPT-1, right? And so, this is a kind of like, the time is now to start making some of these early models, and the hope is the data will catch up. So one of the ways that our lab contributes to the data scaling problem, as I mentioned earlier, we have all these perturbation libraries. We have decreasing, library preparation methods. And so that allows us to do basically scalable perturbation screening. And perturbations really allow for a combinatoric expansion, right? If 60 million cells is all the observed cell states in the human body, but now you can cross that with 20,000 gene knockouts, 5,000 transcription factor overexpressions, right? You start to really build on that complexity. So an example I'll show for what our virtual cell can do. I'm not going to get into the model or math today because we're not ready to share that, but I'll just show like an example of something we can do. Perturbation prediction is something we hold near and dear to our heart because we really want to be able to do in silico drug screening. And so the two types of things you might want to do are you have drugs in different cells, and you have a new cell type that you only have wild type data. Let's say you have, liver fibrosis single cell sequencing from human donors, right? you can then project the drugs into those human samples and see what the drug would do to those people. The other alternative is you have some perturbations in all the cell types you care about, but you want to predict new perturbations that you haven't seen before. And that's hard, but also doable. To give an example of what we can do, here's where we're treating immune cells with interferon gamma, 4 different immune cells for each plot. And the control cells are shown in green, the perturbed cells are in teal, and the predicted are this dark blue purple. And you can see that in general, our model is able to actually predict the cell types, where they should be in this UMAP space and what their transcriptome should be. We're also developing a lot of other evals, but I just wanted to show a little preview of how this model can actually work. And I'll just show also an example of the power of this. So we did an example where we took muscle rejuvenation, muscle aging single cell data from humans. This is a paper last year. They had 8 decades of life, single cell sequencing of the muscle from the people. And then we screened thousands of drugs in silico on the muscle to ask, can we make old cells shown in green here look young again, shown in teal? And so one drug, actually made the cells go in between. This is this blue population here. And it was this molecule genistein, which we had never heard of. It's an isoflavonoid from plants. You can actually buy on Amazon. They sell it for like women's health, I think, for like boosting your immune system. But it turns out, our models suggest it might be useful for muscle rejuvenation. And we actually found the paper. It is an old paper that really was unnoticed, but there's a paper that says genistein promotes skeletal muscle regeneration through a microRNA pathway. And so we were pretty excited because maybe there's something real here and something we can go validate in the lab, which we're looking at. So with that said, we're very interested in, for example, building these virtual cell atlases where we can do all these in silico genetic screens, drug screens, you can imagine across every aging data set that's out there, every disease data set that's out there, and uncover hopefully new targets, pathways, and understandings of these diseases. So, in general, we're very excited about building out the virtual cell. We'll hopefully have some of our first papers out on this year. We're doing a lot with, not just drug screening, but also tox prediction, building this stuff into digital tissues and even humans, and really just building an end-to-end framework from, health data, molecular data, biomarkers, and, hopefully having like essentially a foundation model that can project all of this into outcomes for people. That's our, I think the overall vision. And, if in 10 to 20 years, you could basically have a disease, you sequence someone, you have their clinical data, and you could predict drugs for them and, be testing this in the phase 1 within months, I think that would be an incredible future. We'll see if we can ever get there. But with that said, we have many teams in the lab working on these various projects, and of course, funders sort of backing the lab. So yeah, I guess with that, we can maybe switch to questions.
Yes, thank you for that wonderful presentation. Yes, can you, yeah, okay, the screen is visible, I think. So yeah, we do have indeed quite a couple of questions for you, because this was very, very insightful and yeah, very, very intriguing as well. So maybe let's start with the Evolve Pro model, which you mentioned. Do you think this model can also be applied to more complex proteins in terms of size and quaternary structure?
Sorry, with regards to like what about the quaternary structure?
Like, do you think you can apply the Evolve Pro model for proteins that are larger in terms of size, or more complex in terms of the structure, like for example multimeric proteins?
I see. Yeah, I mean, I don't think the protein size should be limiting. Because really the language model foundation, the foundation language model should really be able to capture small or big proteins and allow us to infer where to mutate. On complexes, that's an interesting question. Yeah, I mean we haven't tried complex evolution. I would think if you actually built you would have to do some modifications to Evolve Pro, but I think if you co-embed the different proteins that are a part of your complex and you evolve them together, it should theoretically be possible. Yeah. It's an
Thank you.
interesting thing to look at. Yeah.
Yeah, and then another question regarding Evolve Pro. Considering it's designed for protein evolution and engineering, but do you think you could use this model or a similar strategy to develop a method for nucleic acid therapeutics, such as ASOs or aptamers?
Yeah, I do think actually you could. It's something we've been thinking about, but like in maybe the past 6 to 12 months, there have been some really good DNA and RNA models that have been developed. So as long as you start with like an underlying foundation model for the sequence you're trying to embed, this approach should work. And so if we're doing like mRNA based optimization, I think you could take one of those like RNA BERT like models, like codon BERT or something, and apply the same Evolve Pro framework, and it should work.
Thank you. Yeah, we have another question regarding Evolve Pro. Can you tell something about this Evolve Pro LLM, like how it was trained, for example?
Yeah, so we don't do like canonical pre-training in the same way you would with like a foundation model. So we basically what we do is we build a top layer model, which in this case random forests actually perform the best even on top of like MLPs and other neural network architectures. But you basically you can start with either zero-shot sequences or random sequences. You go test them across your assays and you feed that back through the embedding and then along with the assay data into the random forest, and it can then predict essentially the next set of mutations to do. Um, and that seems to work pretty well. It can pick single mutations, and later on in evolution, it can start combining single mutations it's seen to make multi-mutants. And we're actually working on sort of next generation versions of this that are even more generative, so you can move beyond single or triple mutations to many more mutations. It uses a much more complex set of algorithms to basically reason about where to mutate in the protein. But those approaches seem to be working pretty well, and we'll hopefully have another paper on that soon.
Yeah, we have another question here. Do you see these foundational cell models being used to better understand disease pathogenesis, like how specific gene or environmental perturbations drive cells towards a disease state?
Yeah, so that's actually we want to make like a disease virtual cell atlas where we basically do in silico gene knockouts and gene overexpression across like the 160 diseases that are in cell X gene, for example. You could use that to infer, what perturbations make wild type cells look like diseases and what make disease cells look like wild type. And a lot of that could point you to interesting targets or pathways to understand the disease better.
Thank you. Yeah, we have another question regarding Evolve Pro. I think it relates back to one of the earlier questions when I asked about the nucleic acid therapeutics. This one is targeting more nucleic acid enzymes. So can Evolve Pro be used for the nucleic acid enzyme evolution?
Nucleic acid enzyme evolution. Um, so like, do you mean like CRISPR enzymes, or what do you mean by nucleic acid enzyme?
This is a question from our audience. So let's see if we get more information,
Okay.
but I would
Well,
think so. Oh, DNA enzymes. Yeah. DNA enzymes.
like DNA enzyme, sure. Yeah, I mean,
Yeah.
so I didn't show this in the paper, but a bunch of the proteins we did were like, we did a mini CRISPR protein that was very inactive, and we were able to evolve it to be the most active mini CRISPR protein. We compared it to all the other existing ones. We took a DNA integrase, like a BXB1, and we evolved it to be better. We actually evolved prime editing as well. So it works with, yeah. We showed, I think, 6 different functional classes in the Evolve Pro paper. Beyond the in silico ones. Like we in silico tested across 12 different proteins in Protein Gym, but in our own hands, in our own lab, to state of the art levels, we did 6 different classes.
Yeah, then another question, not on the Evolve Pro specifically, but more general. You mentioned at the start of the presentation, small insertions, small deletions, but now also large insertions, which can be quite challenging, of course. What about large deletions, like in case of certain neurodegenerative conditions with repeat elements, for example? Do you think that would be an interesting way to go as well?
Yeah, I mean, large deletions, I think maybe not fully solved, but people have shown with like prime editing you can do deletions of like thousands of base pairs. I think it's not super efficient, right? Is one of the issues. So if you could find a way to make it more efficient, that would be great. It relies, of course, on like all these DNA repair pathways that are just so hard to manipulate, and to, if it's just not active, it's hard to boost that. That's, I think, one of the reasons why these DNA editing approaches don't work well. And now you're talking about, okay, for neurodegenerative disease, you have to deliver this to the brain, and there you take a hit as well, and it just becomes difficult, which is why I like the trans splicing approach a lot more. But yeah, I mean, you could definitely, you think with deletions, you can't just delete like exon 1 of Huntington's also. You need to replace, and so the delete and replace has to happen, which adds another layer of complexity. Um, but yeah, maybe there's other stuff you could think about doing deletions for. I can't really think about
Yeah.
why you would want to delete something, to be honest. Yeah, because if you're deleting it because it has a mutation, why not just edit the mutation? Um, and like, I guess if there's like an insertion, maybe you want to delete the insertion. Like sometimes people have like insertions as their genetic mutation. Yeah.
Yeah. Yeah. Okay. Yeah. And like linking it to like maybe more controversial topic, like for example, chromosomal abnormalities, which is kind of a different scale than just point mutations. Yeah. Do you think
Yeah.
that's maybe where this could go?
Yeah, perhaps. Yeah. Long way to go though to make those levels of giant like mega base level edits. But yeah.
Yeah. Okay, great. Yeah, regarding the last part of the presentation you mentioned, so you're building this virtual cell, which is very interesting. But do you think you could maybe also eventually, if you have sufficient computing power, build something like a virtual tissue and eventually a virtual organism? Is that kind of the goal there, or
That's
how
what
do
we're
you see this
building
evolve?
towards. Yeah, that's absolutely the goal.
Yeah, yeah,
We're very close to being able to do this. The virtual tissue, I think, is actually easy. The virtual human requires not just like the buildup of those things, but other types of data input that we've been working on to get. But um, yeah, I think that's absolutely the vision. So
yeah. Very cool. Yeah. Oh, yeah, we have another question here. How big of the insertions can be introduced by trans splicing?
we show in the preprint, I think it was up to a few thousand bases. We only tested up to a few thousand bases, but uh, there's really no reason you should be limited. I mean, like splicing deals with like tens of thousands of base pairs, right? So theoretically, you should be able to do longer lengths, but with our systems, you start taking delivery hits, right? Because the longer you're putting something in, you have to actually get that into the cell. Um, so, but yeah.
Great. Yeah, and then going back again to the final part of the presentation where you showed this muscle rejuvenation model. Have you evaluated this also at the functional level?
Um, we have not yet. We are actually gearing up for it. We just set up the models in the lab.
Yeah, sure.
So the hope is we'll have experimental validation. We're also, I mean, I'm just showing like one example of an output. Like with our newer models, we get all sorts of predictions, and the goal for the paper is to validate at least some fraction of them. With other types of AI models, my goal is like 5 to 10% accuracy to start, right? Like if you can make, maybe like 50 predictions and like, 5 to 10% of them are correct, that's pretty amazing because, we're doing predictions like for like new types of regenerative therapeutics, and it'd be very hard to come up with that otherwise. And so, well, yeah, but we'll see how good they are. It's like with de novo protein designs, like you have to screen hundreds of designs and maybe like one of them will work, right? Or like 10 of them will
Yeah,
work. But
yeah,
that's like, yeah, that's the state we're at right now.
yeah. Yeah. Okay. Thank you for explaining that. Yeah. So I take it that because that was actually a follow-up question. If you have any examples of models that you have actually validated at the experimental level, but I assume indeed the accuracy may be something to yeah, that
Yeah,
may be
well,
something to improve on first.
like, yeah, I mean, we haven't published this yet, so we'll have
Yeah,
it.
sure.
But like, if you want an example, like Google had their AI scientist, um, published a couple weeks ago, and I think they used it to predict like some new cancer drugs, and I think some of them seem to have some, proliferation, an inhibition and proliferation effect on the cancer cell lines, right? Or I think GeneFormer, this is a transformer model that was trained on single cell data, that was published a couple years ago. They predicted some like heart, I think arrhythmia drug or something with heart, and they tried to validate it in some in vitro model, and they seem to have some effect, right? So there are examples where these types of models can work. So I feel pretty good that there will be some fraction of things that'll work. Yeah.
Yeah, and then one more question regarding the fact you're working on neurodegeneration, so you're focusing also a lot on blood-brain barrier crossing, I guess, and uptake. Have you thought about applying these AI models that you developed to predict, for example, the blood-brain barrier crossing or permeability of certain drugs?
We have not, but it is a task. So we have like our wish list of foundation model tasks, and blood-brain barrier prediction is one of them. Yeah, there's complexity in how you would set it up, because that's like a tissue level task, right? And so there's like
Yeah.
questions of what cell types maybe do you predict on, and like what actually matters, but it's something we'll look at. Yeah.
Great. Okay. I think that's it for the questions right now. So I'm gonna hand the floor back to Kathy. But yeah, thank you so much. That was very insightful. And yeah, it was a great presentation. So thank you very much for that. And then
Yeah,
Kathy,
thanks for
please
having
go
me.
ahead.
Yeah, thanks, Dr. Abudayyeh and Sarah. Please know that Creative Biolabs regularly invites estimated experts in life science and biotechnology to share their latest findings in our webinar series. We encourage you to visit our website and subscribe to our social media channel so you won't miss any upcoming events. And that is for today. And thank you everyone for joining us, and we will see you next time.